Let's begin our discussion of the organelles with the nucleus. The nucleus actually has a double membrane, but it's a different double membrane than the mitochondria and the chloroplast. The nucleus is not believed to have arisen through endosymbiotic means. Instead, when we look at this doubled membrane, what we see is that the two membranes connect with each other. They actually come in contact with each other at particular locations across the surface of the cell. Those locations are called pores. Now, the name pore makes it sound like it's an open place and it's always open 24 hours a day. But in reality, nuclear pores are very complex. They are considered gated. In other words, there are physical structures that block the pore under most conditions so that only certain molecules are allowed through the pore and into the nucleus or back out of the nucleus. So the nuclear envelope, while it does have two layers in it, those layers are physically connected to each other. They're joined together wherever there's a pore. That's why we call it an envelope. If you think about it, the envelope that you would put a letter into, it has two pieces of paper on top of each other. It is a doubled piece of paper, but the paper is actually connected around the seams of the envelope. The same thing is going on in the nuclear envelope. The connections are in the areas of the pores. Now, the other structure that we should be familiar with as we discuss the nucleus is a structure that we sometimes see under the microscope, and it's this dark area right here. So we are looking at a cell. We are actually looking at a TEM image, a transmission electron microscope image, and we are looking at the nucleus right here of this eukaryotic cell. So most of this material in here that you can see is chromatin, DNA wrapped around histone proteins. This region right here that stains so darkly, this is called a nucleolus. And what a nucleolus is not so much a physical structure as a functional structure. This is an area inside the nucleus where RNA is being actively synthesized. So wherever you see these dark regions, you know that a process called transcription is going on in that area at that moment. Transcription is the name of the process where the DNA molecule is being used to make an RNA molecule. The nucleolus can be singular, like this one, and sometimes you can see several of them in a very active nucleus. Next on the list of the organelles is the mitochondria. Now, the mitochondria is also an organelle with two membranes, but the mitochondria has a true double membrane. In other words, those two membranes are separate from each other. There's what we call an outer membrane and an inner membrane. Now, the inner membrane is actually larger in terms of its surface area than the outer membrane is. So when you take a larger membrane and you fit it inside a smaller membrane, you end up with folds in the inner membrane. One of the analogies that I like when I'm thinking about the inner versus the outer membrane of the mitochondria is sandwich bags. You know those plastic baggies that you can put your sandwich in? You can buy them in different sizes. You can buy a gallon size bag. You can buy a quart-sized bag, you can buy a sandwich-sized bag. Well, imagine if you took a gallon-sized baggie and you put it inside a sandwich-sized baggie. The gallon baggie is the only way it's going to fit is if it folds up, if it wrinkles up and folds up so that it can fit inside that smaller space. That's exactly what's happening with the mitochondria. The gallon baggie is the internal membrane, and the sandwich baggie is the external. So the external membrane is nice and smooth to our eyes under the microscope, and the inner membrane is all folded up into structures that we call crista. Now, recall how mitochondria are believed to have evolved. We believe that a larger primitive cell engulfed a smaller primitive cell, and that smaller cell became the mitochondria. Remember, that's what we call endosymbiotic theory. The result to having these two membranes is that mitochondria have several different compartments inside of them. The space, for example, between the outer membrane and the inner membrane, that space right there, is referred to as the intermembrane space. That's a good name for that space. That's a name that makes a lot of sense. Inter, which means between, membrane space. So the between membrane space. Now, if we look inside the internal membrane, so the cavity that's inside that larger baggie, that gallon-sized baggie, that's a cavity that we refer to as the matrix. The matrix is full of a material that's a lot like cytoplasm in that it's quite gel-like. The matrix is also the place where you find the mitochondrial DNA molecule. So we shorthand that as mtDNA. Mitochondria, if you remember, are the organelles inside of eukaryotic cells where the process of ATP production occurs. So the process that produces ATP is referred to as respiration, cellular respiration. And in organisms like you and I that are dependent upon oxygen, that is an aerobic process. It's an oxygen-dependent process. Aerobic cellular respiration involves a simple sugar, glucose, being systematically broken down and the energy harvested from it so that the molecule called ATP can be produced. And of course, ATP is the molecule that cells use for energy. You should also note that ATP production is not the only thing that occurs in mitochondria. Some of the oxidation or the breakdown of fats also occurs in the mitochondria. The other thing to know is that the number of mitochondria that we'll find inside different types of eukaryotic cells is going to vary. In other words, some cells are going to have a lot of individual mitochondria in them, and other cells are only going to have a few. It all depends upon the energy needs of that type of cell. If you're looking at, for example, a skeletal muscle cell, and again, we call those fibers. If you look at a skeletal muscle fiber, it's going to be loaded with mitochondria. And that's because the process of skeletal muscle contraction is very energy intense. It just requires a lot of ATP. Other cells, like for example, the cells that hold fat, those cells are not particularly metabolically active, and so you'll only find a handful of mitochondria, if you will, in a cell like that. It all depends on the energy needs of that particular cell, how many mitochondria will be there. Next on the list is the chloroplast. The chloroplast, like the mitochondria, is a true double membrane organelle. It has an outer membrane and an inner membrane, and those two membranes are separate structures. Chloroplasts, like mitochondria, are believed to have evolved through endosymbiosis. Now, if you look at this drawing up on the upper right-hand side, the artist has drawn for us this outer membrane in a light green color, and then an inner membrane in a darker color. What's hard to see in this drawing, though, is that this inner membrane is also very folded up in there. It's just not folded up the same way that we saw the inner membrane folded up in mitochondria. If you recall, in the mitochondria, it's just a larger surface area being forced into a smaller surface area. So you get these just basic folds in that inner membrane structure, those crista. If you look at the chloroplast, the folding is much more organized. It's much more highly organized. The folds of the inner mitochondrial membrane create these structures, these stacks of what look like pancakes. These stacks are referred to as grana or thylakoids. In fact, we refer to the inner chloroplast membrane in general as the thylakoid membrane. So there's an outer membrane and there's a thylakoid membrane. And the thylakoid membrane is folded into these pancake stacks. Now, just like we saw with the mitochondria, the chloroplast also has compartments within it that we need to be familiar with because different processes are going to occur in each different compartment. So the space between the outer chloroplast membrane and the thylakoid membrane, the space in between the two, is referred to, again, as the intermembrane space. Easy to remember. What is essentially analogous to the matrix in the chloroplast is called the stroma. So the stroma is the space within the inner membrane, the cavity that surrounds these grana. Next on the list of organelles is the endoplasmic reticulum. The endoplasmic reticulum is the first of the single membrane organelles. If you translate that term, endoplasmic reticulum, the endo portion of it means within. So within the plasm, in other words, within the cytoplasm, and reticulum means mesh or network. So this is a network or a mesh structure that when you look at it under the microscope, it looks like flattened sacs of membrane. The endoplasmic reticulum is actually continuous with, in other words, attached to, the outer membrane of the nuclear envelope. That's going to be important for us to recall later on when we think about how things move between organelles in the cell. There are actually two varieties of endoplasmic reticulum in a eukaryotic cell, and you may have heard of these. The first is called rough endoplasmic reticulum, or RER, And the second one is called smooth endoplasmic reticulum, or SER. Now, the difference between these two is whether or not there are ribosomes located on the outside of the structure. So the rough endoplasmic reticulum got its name because when scientists examined it under the microscope, the surface of this organelle looked very rough, bumpy, in other words, lumpy on the outside. And the reason for those bumps, the reason for that rough surface, is that there are a whole bunch of ribosomes located on the outer surface of this organelle. And remember, ribosomes are involved in protein synthesis. So that tells us what the rough endoplasmic reticulum is involved in. It's involved in this processing that goes on with proteins that are produced by the cell. The proteins that are produced on the ribosome through translation are actually brought in to the endoplasmic reticulum, the rough endoplasmic reticulum, and they are processed, they are amended. Bits and pieces of them are taken off, things are added on, they are carefully folded. This is the kind of processing work that occurs inside the rough endoplasmic reticulum. The proteins will travel through all of this network of membrane through the rough endoplasmic reticulum and then travel on to other parts of the cell where they will be further processed before they go to wherever it is that those proteins are going to live and function. All of this interconnected membrane and interconnected network between the organelles gets a name. We call this the endomembrane system, and I'll speak more about the endomembrane system in just a minute. But first, I don't want us to forget about the smooth ER. The smooth ER under the microscope, like the name suggests, looks very smooth on its surface. There are no ribosomes sitting on the smooth ER. And that tells us something. The smooth ER has nothing to do with protein synthesis. Instead, what's going on in that organelle is the synthesis of lipid and sterol specifically. The other thing that happens inside of the smooth ER is the detoxification or the inactivation of certain types of drugs. There are certain drugs that are dismantled or metabolized, if you will, inside the smooth ER. On this slide, you can see a drawing of the endoplasmic reticulum. And the reason I like this drawing is because it shows how both kinds of endoplasmic reticulum are actually attached to each other. And the rough endoplasmic reticulum is actually attached to the outer layer of the nuclear envelope. So if you look over here where you see this purple color, this represents the nucleus. And remember, the nucleus has a double layer of membrane called the nuclear envelope. Attached to the outer part of the envelope is this red structure. This is rough endoplasmic reticulum. This is a big network, a big maze of flattened compartments of membrane. So you could go from the outer membrane of the nucleus, you could travel into this maze of compartments. You could pass here, and then you could travel here and through here, and that's exactly what proteins do. They travel through the rough ER, and they go through all of this processing that occurs. Now, the rough ER is continuous with the smooth ER, which is not to say that proteins go through the smooth ER. They don't ordinarily. Lipids are being synthesized in here. Drugs are being detoxified in here. But the membrane is all connected. It's part of, again, what we call the endomembrane system. The other thing I like about this drawing is how the artist has shown us that the compartments in the rough endoplasmic reticulum really do look like flattened sacs. Compare that with the compartments that are in the smooth ER. The smooth ER is much more tubular than the rough ER is. It has a much more tubular look to it. To my eyes, it almost looks like coral compared to these flattened compartments in here.